Welcome back, everybody. In this second part dealing with meiosis, we're more specifically dealing with how we actually are unique. In the previous video, we discussed about how meiosis takes a diploid cell and creates 4 haploid cells at the end. But one of the things that's different about meiosis compared to mitosis is that each one of these cells that are produced is actually unique. So mitosis is cloning. Mitosis is taking one cell and creating two identical copies of that cell. The entire point of meiosis is to make them not identical. We don't want all your sperm cells to be the same if you're a guy, or all your egg cells to be the same if you're a female. We want this variation. That's the entire point of sexual reproduction, is to create this variation, to create these differences, to increase the genetic diversity of our species. So this video will go through the two different ways meiosis uses to create that genetic diversity, and also wrap up going through some of the similarities and differences between mitosis and meiosis. Meiosis produces genetic variation in two different ways. The first way deals with random assortment of chromosomes, this idea of random assortment, which is best exemplified through an example in some diagrams. During metaphase 1, we talked about how the chromosomes line up in this double file, and how the homologous pairs are actually what are lining up. And remember, within each homologous pair, one came from mom and one came from dad. And we're going to assume that your mom and dad are not the same person, so therefore that they're very similar information but slight differences. Maybe mom's chromosome has blonde hair while dad's chromosome has the gene for brown hair. Well, when these chromosomes line up, so let's say that dad is blue and mom is red, when these chromosomes line up, you can see that if we only had 4 chromosomes, one possibility is that dad's blue chromosomes actually line up next to each other, and mom's red chromosomes also line up next to each other. But it's also possible that these two could flip-flop. That on one side you could have mom chromosome, and the next chromosome could be a dad chromosome, and on the other line you could have a dad chromosome and a mom chromosome. So each of these is possible. There's no set order. It doesn't say that dad's has to go on the left, mom's have to go on the right. Each one of these pair can flip-flop and can go in either orientation. And then if we look at metaphase 2, the entire point of to separate these chromosomes, so meiosis 1 is going to separate these pairs. So the 2 left will go to the left cell, the 2 right will go to the right cell. And then during metaphase 2, they'll line up single file, and they'll get ripped apart. Single file get ripped apart. And we can see here that we could create a cell that is all blue, a cell that is all red, a cell that has the big blue chromosome and a small red chromosome, and a cell that has a big red chromosome and a small blue chromosome. So you can see that with 4 chromosomes, you can, by this random assortment, by this flip-flop arrangement, you can actually create 4 different combinations. Well, we have 23 pair of chromosomes, each that can flip-flop in whatever orientation they want. So we can actually create 2 to the 23rd power number of combinations based strictly on random assortment alone, which is a massive amount of variation just by this random assortment of chromosomes, just by this alignment of chromosomes during metaphase 1 creates 2 to the 23rd power number of different sex cells in a human. So this is a very powerful mechanism to create variation in your gametes, in your sex cells. The second way that we create variation is through crossing over, which is when there's an exchange of genetic material between homologous pairs. And this occurs during prophase 1. So we discussed that during prophase 1, the homologous pairs pair up. So dad's chromosome number 1 finds mom's chromosome number 1. And when they pair up, sometimes they get a little too frisky with each other and can actually get tangled up, as you see down here. And depending on which way we cut and release them, either they can still maintain their normal DNA structure and DNA sequence, or sometimes if we cut them in the wrong orientation, we can actually see that they can flip and swap DNA segments. So we can create new combinations that have never been seen before through this crossing over event, where a little bit of mommy's chromosome gets on daddy's chromosome, and a little bit of daddy's chromosome gets on mommy's chromosome. And then if you actually were to look at this, so originally this is the original cell with one set of each chromosomes. Then during the S phase, we duplicate, so now we have the full X-shaped chromosome. During prophase 1, they pair up, and there's a little bit of crossing over. depending on which way we cut them. Let's say we cut them improperly. So now we have a little bit of each. And now during metaphase 2, each one of these chromatids will get separated and go into a unique cell. So we can see that we have one cell that has ACE, one cell that has ACF, one cell that has BDE, and one cell that has BDF. So once again, this crossing over has created even more variation. And this combination between the random assortment of chromosomes and crossing over creates all of the variation you see in your sex cells, that none of your sex cells, none of your gametes, no two sperm cells, no two egg cells should ever be the same in your body. And the second part, I just want to briefly go over some and highlight the differences and similarities between meiosis and mitosis. One of the big things about meiosis we talked about is that meiosis 1 is all about separating these homologous pair of chromosomes, while mitosis is all about separating the individual chromatids. So meiosis 1 is separation of homologous chromosomes. Mitosis is the separation of the individual chromatids. And then during meiosis 2, since it is the same as mitosis, meiosis 2 is separating those individual chromatids from the individual chromosomes. Comparing them side by side, no matter what, before mitosis or meiosis happens, you have this DNA replication, so you have this S phase. No matter what interphase is occurring before the M phase. Don't care if it's mitosis, don't care if it's meiosis, interphase is still happening. In mitosis, there's only one cell division. Those cells start out as diploid, they end up as diploid, so you still have two end cells. In meiosis, they start out as diploid. The first cell division, meiosis 1, creates haploid cells, and then meiosis 2 is just taking those cells, dividing them further, and creating a total of 4 haploid cells at the end. And this pretty much just shows what I just actually drew. So meiosis starts with a diploid cell, DNA replication happens. Meiosis 1 is this reduction division going from a diploid to 2 haploids. Then meiosis 2 is just taking those 2 haploids and creating even more cells. So you have a total of 4 that are produced. While mitosis is taking a diploid cell, DNA replication happens during S phase, and then it's only one division, and they're identical copies, so it's a diploid to a diploid. And to summarize the differences, meiosis always is producing 4 daughter cells that have half the number of chromosomes, so they're haploid, while mitosis only produces 2 daughter cells, and they're identical, so they have the exact same number of chromosomes as the parent cell. Meiosis produces daughter cells that are not genetically identical, so you want that variation, you want those differences, while mitosis wants to create clones. So mitosis is cloning, it's creating identical cells. And the last thing to compare them, meiosis has 2 cell divisions, while mitosis only has 1 cell division. And those are the big differences I expect you to know between mitosis and meiosis, both how many cells are produced, how many divisions occur, are the cells haploid, are the cells diploid.